the study of metabolism became a science based completely on chemistry, physics, and mathematics. Metaphysics and mysticism were eliminated. Apparently, all parts of a plant except for seeds store minerals in soluble form in the central vacuoles of cells. Nitrogen can be concentrated a little by being converted to compounds with multiple amino groups such as asparagine, citrulline, and so on, the same compounds that are used in nitrogen transport. Phosphates, sulfates, and other mineral nutrients apparently are simply sequestered in the central vacuole in the same forms in which they are used metabolically by the cell. This method does not permit storage of large amounts of minerals because high concentrations would be toxic, but plant cells differ from our cells by having very large vacuoles and just a tiny amount of cytoplasm squeezed between the vacuole membrane and the cell membrane. Pound for pound, plant tissues need only very small amounts of minerals compared with animals. In contrast, seeds do store minerals. Seeds must be both lightweight but also packed with enough resources to get a seedling established quickly. Amino acids are stored as particles of protein packed so tightly that it typically crystallizes into a structure called a protein body. In many seeds, protein bodies themselves contain inclusions of another crystalline form, crystals of a substance called phytin. Mu-inositol hexaphosphate is a 6-carbon sugar that has 6 hydroxyl groups, each carrying a phosphate bound to it. Being an acid, mu-inositol phosphate ionizes and loses protons, H, when dissolved in water, but when the developing seed concentrates it and precipitates it in the protein body, it puts cations on it rather than returning the protons, and the cations used are Mg2+, Ca2+, Zn2+, and K. This mineral holding form is phytin, and it permits the protein body to not only store amino acids and phosphate but all of these other essential elements as well. Figure 13-19 A cultivated ant plant, Hypnophytum, with a greatly enlarged tuberous hypocotyl. Holes visible on the surface form naturally and give ants access to chambers inside the hypocotyl. A young plant of Hypnophytum, cut open longitudinally to show the chambers that develop naturally inside. Because this is cultivated in a greenhouse that controls insects and other small animals, no ants live in these chambers. The enlarged hypocotyl of another ant plant, Myrmecodia, many small entrance holes are visible. This fern, Lechinopterus carnosa, is also an ant plant, having swollen, hollow chambers in its stem. Figure 13-20 Plants of Dischidia major are also ant plants. They form 2 types of leaves, some are cup-like and clasp a tree trunk or branch, others are hollow, balloon-shaped leaves. Ants live in both types and debris accumulates. The Dischidia stem sends adventitious roots into the debris trapped by the leaves, absorbing nutrients from it. This plant is cultivated in a greenhouse too clean to have ants. Plants and people. Box 13-4 Fertilizers, pollution, and limiting factors. Plants in nature usually do not grow as vigorously as they potentially could. For example, desert plants typically grow more rapidly if given extra water, plants in shady areas grow better if given a bit more light, and prairie grasses, which already have enough water and light, benefit from extra nitrogen fertilizer. Any plant grows at a particular speed and vigor because it is limited by some factor, such as too little water or light or nitrogen fertilizer. An important concept is that there is only one single limiting factor at a time for any plant. Desert plants given more light or fertilizer will not grow faster, it is water that is limiting them. But if we do give them extra water, their growth rate will increase until some other factor becomes limiting, perhaps lack of nitrogen. While growing slowly, the plants could get nitrogen quickly enough, but now that they are growing faster, their ability to obtain nitrogen from the soil may be limiting. If we give the plants both water and nitrogen fertilizer, they may grow even more rapidly, but some other factor will become limiting. On many farms, plants are irrigated and fertilized and planted far enough apart that they do not shade each other and their roots do not interfere with each other. Such plants grow much more rapidly than they would in nature, but they are still limited, in this case, by their own genetics, their own innate metabolic capacity. There is always a limiting factor. The concept of a limiting factor is important in understanding techniques for reducing the damage caused by pollution. Under natural conditions, the water in rivers and lakes has so few nutrients that algae grow slowly, and they are so sparse the water is blue. In the mid-20th century, pollution from farms and cities fertilized rivers and lakes and allowed algae to grow more vigorously. As rain drained from fields, lawns, gardens, and golf courses, it carried much of the fertilizers that had been applied to stimulate the growth of crops, flowers, and grass. Also, most household waste that is flushed down toilets is an excellent organic fertilizer. With all these extra inputs of nutrients, populations of algae became so dense that pond scum floated near the surface of rivers and lakes, and the water was green because of the abundance of microscopic algae. It is difficult to stop all pollution, but it is only necessary to control one single pollutant to create a limiting factor. Phosphate was chosen as the target. Phosphorus is an essential element, and it is naturally low in pure water. A large amount of the phosphate pollution in rivers comes from laundry detergent and dishwashing soap. With a little effort, phosphate-free detergents were invented, and now they are used almost universally, so there is much less phosphate pollution. The concentration of phosphate in rivers dropped so low that algae could no longer thrive, and their populations fell to more normal levels. Even though the water is still heavily polluted with nitrates, sulfates, and other nutrients, the algae cannot use them as long as phosphate is kept low enough to limit their growth. If we could reduce the phosphate runoff from farms and lawns, the levels of algae would drop even more and rivers and lakes would be even cleaner. It would be better to control and reduce all types of pollutants, but by keeping one at limiting levels we can at least minimize some of the damage caused by pollution. Figure B13, for this river runs through extensive agricultural areas in East Texas and is polluted by fertilizers from fields and manure from livestock. Because the water is so rich in nutrients, algae and bacteria grow well, giving the water a brown color. This stream in North Carolina runs through a forested area without agriculture, so it does not receive as much pollution as the river in. There are many towns and homes along this river, but because modern detergents do not contain phosphates, the wastewater dumped into this river is almost phosphate-free, so algae and other microbes cannot grow in it, and the river has clear, blue water. At the next level. 1. Composting. Many people are trying to live more harmoniously with the other creatures on Earth and are trying to reduce the harm we do. One aspect of this is to use natural, organic fertilizers rather than artificial, inorganic commercial fertilizers. Most organic material must be composted before it can be used. In composting, the materials are collected into a large pile and allowed to ferment for weeks or months. Enzymes and microbes break down complex molecules, freeing the minerals. The fermentation causes the compost pile to become hot enough to kill harmful bacteria, which is important if the compost contains manure or animal parts. The process of composting is easy to do at home and it involves complex biology, so it is an excellent topic for independent study at the next level. 2. Mineral nutrition and climate change. Increased greenhouse gases cause the oceans as well as the land to become warmer. This causes more oceanic evaporation, which later falls as rain. Every time rainwater runs off into streams and rivers, it carries some minerals with it, washing them out of the soil. If the area has a large amount of dead plant material, for example as leaf litter and humus, this helps to retain the minerals, but desert areas have little organic matter in the soil, and when they receive more rain, they lose more minerals. As earth warms and rains increase, soil leaching will also increase, affecting the fertility of the soil and ecology of regions. A key word is biogeochemistry. 3. Hydroponics. Hydroponics is the technique of growing plants without soil. Originally, plants were grown with their roots in aqueous solutions of dilute fertilizer, but now some techniques involve merely misting roots periodically to keep them moist and nourished. By growing plants hydroponically, we can control their water and fertilizer precisely and eliminate soil-associated pests such as nematodes. For space exploration, using hydroponic misting allows plants to be cultivated without the weight of either tanks of water or soil. Experiments are being performed on the International Space Station. Hydroponics will be especially important when we send manned flights to Mars and must not only grow some fresh food but also recycle water and reuse human urine and feces by means of composting. Some of these problems are discussed in the book Packing for Mars, The Curious Science of Life in the Void by Mary Roach. I strongly recommend this book as an enjoyable, humorous, and informative introduction into the problems of biology in microgravity. Summary. 1. Plants synthesize all their body's materials using only carbohydrate and a few essential elements. 2. 3 criteria must be met for an element to be considered essential. It is necessary for the completion of a full life cycle, it cannot be replaced by a chemically similar element, and it must act inside the plant. 3. Macro or major essential elements are needed in relatively large amounts, whereas micro or minor or trace elements are required in only extremely small amounts. 4. Deficiency diseases are rare in nature, probably because susceptible plants are outcompeted by deficiency-tolerant plants. However, deficiency symptoms are frequent in crops and horticultural plants if proper fertilizing is not maintained. 5. Deficiency of an immobile element produces symptoms in young leaves and buds, whereas deficiency of a mobile element results in symptoms in older organs. 6. Cations are released from the surface of soil particles if protons, which result from the respiration of roots and soil organisms, disrupt their electrical attraction. 7. Mycorrhizae are the principal means by which most plants absorb phosphorus. 8. Nitrogen metabolism consists of fixation, reduction, and assimilation. 9. Nitrogen must be obtained from the air, but only certain prokaryotes, some free-living, others symbiotic, have the necessary enzyme, nitrogenase. 10. Most plants absorb nitrate, which must be reduced by the plants, using large quantities of NADH, NADPH, and ferredoxin. 11. Once formed within plant cells, ammonium is assimilated by transamination, being passed from glutamate to various amino acids. Important terms. Necrosis, nitrifying bacteria, nitrogen assimilation, nitrogen fixation, nitrogen reduction, trace elements, transamination, weathering. Review questions. 1. Most elements that are essential for plant growth are present in the. How do the elements become available? 2. What is meant by the microflora and microfauna of the soil? Why are they important? 3. How many bacteria typically occur in 1 gram of soil? 4. What is a hydroponic solution? After a solution of known composition which supports plant growth is found, how would you go about testing whether all of the chemicals in the solution are necessary for plant growth? 5. It is simple to grind up a plant and then extract and measure all of the chemical elements present, but this does not tell us which elements are essential. Why not? 6. List the major or macro essential elements. Why are they called that? 7. List the minor or micro essential elements. Why are they called that? 8. Are our modern reagents so pure that we can prepare a hydroponic solution that contains only the 9 major and 7 minor essential elements and nothing else? Can we be certain we have discovered all the minor essential elements? 9. What are the 3 criteria an element must meet to be considered essential? 10. Do you think anyone has performed a mineral nutrition experiment that examined the entire life cycle of a giant redwood? A giant cactus? A mistletoe that is parasitizing an oak tree? What would be the problems involved? 11. To be essential, an element must be acting within the plant, not outside it. What does this mean? Discuss the roles of iron and phosphorus in determining which elements are essential for our plant. 12. We humans, like all other organisms, use a universal set of 20 amino acids in our proteins, but we cannot make of these ourselves. How many are plants unable to make? How many do plants need to take up through their roots? 13. The differences in nutritional resources used by plants versus animals is also great. Plants obtain nutrients in the form of or as simple present in the environment. An animal begins with food in the mouth, but the nutrients occur as in complex which in turn are parts of. 14. Name the essential element involved in each process, changes the osmotic potential in guard cells as they swell or shrink, present in all nucleotides and amino acids, carries electrons in chlorophyll, present in cytochromes, present in plastocyanins. Involved in nitrogen metabolism, present in the water-splitting enzyme of photosystem II, present in ATP, present in the middle lamella, and important in regulating the activity of many enzymes. 15. Under natural conditions, is it rare or common to encounter plants whose growth is disrupted by a scarcity or an excess of mineral elements? Describe 2 habitats that often have excessive amounts of mineral elements. 16. Question 15. Specified natural conditions. What about non-native crop plants and ornamentals? Do they often encounter mineral deficiencies? 17. Name and describe 2 common symptoms of deficiency diseases. 18. What is the difference between a mobile and an immobile element? Where are the first deficiency symptoms for each? Which elements are immobile? 19. Do we understand why certain minerals are immobile in plants? What is one essential element that we humans do not recycle well in our own bodies? 20. What are the 2 fundamental processes of weathering that convert rock to soil? 21. What are size ranges of particles of coarse sand, fine sand, silt, and clay particles? Coarse sand can absorb a great deal of water during a rain, but what happens to the water soon after the rain stops? What would happen instead if the soil consisted of clay micelles? 22. Chemical weathering dissolves the surface of the crystal matrix of rock. As the matrix breaks down, what is released, positively charged cations or negatively charged anions? As a result, the residual crystal matrix has what kind of charge? 23. Many factors affect soil acidity, but probably the most important factor of all is. Describe how this factor exerts its effect. 24. Most soils are too acidic. Desert soils, which have very little leaf litter or other organic matter, are usually. 25. Describe cation exchange. How would decomposition of mulch and humus affect cation exchange? Why is acid rain so damaging? 26. The roots of most plants form a symbiotic association with soil fungi. Name the association and the element that it supplies to the plant. 27. Which essential element is neither a component of rock matrices nor a contaminant in rock? Although this element is abundant in air, no plant or animal can absorb it from the air and use it. Why not? 28. Name the 3 steps in the conversion of N into organic nitrogen that is part of a plant. Which type of organism is capable of performing each step? 29. What is nitrogenase? What chemical does it use as a substrate? It is extremely sensitive to and can function only if is completely excluded from it. 30. The electrons that reduce nitrate to nitrite are brought to it by a short electron transport chain involving F8H and NADH. In which other reactions have you seen these 2 electron carriers? 31. How does nitrogen reduction affect carbon fixation in the stromal reactions? How many molecules of NADPH must be diverted from one pathway to the other? How would respiration be affected? 32. What is nitrogen assimilation? What is the acceptor molecule? What does it react with? What is the product? 33. What is transamination? If an amino group is passed on to oxaloacetate, which amino acid is produced? If an amino group is transaminated onto pyruvate, which amino acid is produced? 34. Figure 13-18 shows several chemicals that carry nitrogen up from the roots to the rest of the plant. What is the unusual feature of these chemicals that make them good carriers? Design credits: Hummingbird, Tango 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Chapter 14. Development and Morphogenesis. Chapter opener image. Every leaf on this wall is facing the same direction, each is turned such that it receives maximum sunlight. This is no coincidence. Instead, individual leaf blades perceive the direction of the optimum amount of light, then transmit that information to cells in their own petiole, causing some to swell, some to shrink such that the blade is rotated and receives optimum illumination. Environmental information has been perceived and transmitted and stimulated a response. Similarly, various parts of a single plant, such as shoots, can generate and send a signal to other parts, such as roots, informing them that more water is needed or less. Outline. Environmental complexity. Light. Gravity. Touch. Temperature. Water. Pests and damage. Responding to environmental stimuli. Tropic responses. Nastic responses. Morphogenic responses. Taxes. Communication within the plant. Perception and transduction. Chemical messengers. Signal amplification cascades. Activation and inhibition of shoots by auxin. Cell elongation. Apical dominance. Differentiation of vascular tissues. Interactions of hormones in shoots. Hormones as signals of environmental factors. Leaf abscission. Tropisms. Flowering. Ripenesto flower. Photoperiodic induction to flower. Endogenous rhythms and flowering. ABC model of flower organization. Box 14-1 alternatives, simple bodies and simple development in algae. Box 14-2 botany and beyond, names of genes. Box 14-3 plants and people, environmental stimuli and global climate change. Learning objectives. After reading this chapter, students will be able to discuss cell communication and coordination in plant growth. Recall the 3 principles of plant response to the environment. Summarize the effects of light, gravity, touch, temperature, water, and damage on plant development. List the 4 classes of plant responses to environmental stimuli. Restate how presentation time and threshold influence plant growth. Recall the names and functions of the most well-studied hormones. List 3 potential cell responses to auxin. Give 2 examples of how hormones communicate environmental change. Summarize how differing photoperiods affect plant growth and flowering. Describe how endogenous rhythms impact plant flowering. Explain the ABC model of flower organization. Did you know? All organisms begin life as a spherical fertilized egg, with differences in genes, gene expression, growth, and morphogenesis producing all the different types of individuals. During growth and development, various parts of a plant send chemical messages to other parts, such that development is coordinated. Organisms obtain information from their environment. If grown in continuous light and unchanging temperatures, plants do not develop properly. Plants grown in microgravity on the International Space Station may bloom and produce seeds, but they often have odd shapes due to lack of gravity. Concepts. All plants begin as a zygote, a single cell, that undergoes enlargement and cell division as it grows and develops into an individual plant. It undergoes morphogenesis and differentiation. Often the zygote divides into a basal cell and an apical cell. This establishes polarity, the formation of the root-shoot axis. Also, the zygote's spherical symmetry changes to the embryo's radial symmetry. Radial symmetry dominates roots and stems, and typically, only leaves, sepals, and other flower parts have bilateral symmetry. Cell differentiation occurs with the first cell division. The basal cell develops a size, shape, and metabolism that differ from those that the apical cell develops. Later, surface cells differentiate into epidermis cells, including guard cells and trichomes. Inner cells differentiate into cortex, phloem, xylem, and pith. Such differentiation requires that each cell be capable of identifying its position relative to other cells, and also of informing other cells of the types of developmental and morphogenic changes it is undergoing. For example, cells that will differentiate into epidermis cells must first detect that they are on the plant's surface. Also, surface cells and roots must develop into epidermis tissue that consists only of pavement cells and root hairs, whereas surface cells of stems of the same plant develop into an epidermis with pavement cells, trichomes, and guard cells. Those of petals might develop into pavement cells only, but filled with pigments. To develop properly, a pre-epidermis cell must not only detect that it is a surface cell, but also which organ it is a part of. Consider also the differentiation of guard cells. They must occur in pairs, and they must coordinate where the stomatal pore forms. It would do no good if each were stimulated to develop as a guard cell, but then each tried to form a pore on one of the sides that does not face the other guard cell. Furthermore, pairs of guard cells do not occur at random on stems and leaves, nor are they clustered together. There is a pattern establishment mechanism that informs each cell of its location relative to other epidermis cells. It appears that after one cell begins to differentiate as a guard mother cell, it produces a substance that moves into surrounding cells and inhibits them from also differentiating as guard mother cells. As the inhibitor spreads, it becomes more dilute until it no longer prevents distant cells from acting as guard mother cells. We now know that the inhibitor is a peptide produced by a gene called epidermal patterning factor-like. The peptide is secreted from guard cell mother cells and diffuses outward into the adjacent young epidermis cells. There, it interacts with another protein produced by a gene called erecta, and the combination of peptide and protein prevents neighboring cells from becoming new guard cells. The peptide cannot diffuse very far, so young epidermis cells that are located far enough away from differentiating guard cells are not inhibited, consequently stomata form with a well-defined spacing between them. Rarely, things go wrong and inhibition is too great. Stomata are then too far apart. Botanists have produced a mutation in Arabidopsis thaliana called too many mouths, in which the pattern establishment mechanism does not work correctly and stomata are formed side by side with few or no pavement cells between guard cells. There are additional control mechanisms as well. For example, if subsurface cells are differentiating into fibers, they inhibit adjacent surface cells from undergoing guard cell morphogenesis. Inner cells of young regions of shoots also must detect their position and differentiate accordingly. Typically, the first cells below the apical meristem to become visibly differentiated are sieve tube elements of protophlome. They are located partway between the epidermis and the center of the stem, and each develops several micrometers away from other newly forming sieve tube elements, like guard mother cells. Each region that begins to differentiate as phloem must inhibit adjacent regions from also becoming phloem, thus establishing a pattern of one ring of bundles in most seed plants, numerous bundles in monocots. Vascular bundles always contain a strand of xylem located just interior to the phloem. Thus, the first sieve tube elements may be capable of stimulating another pattern, xylem formation. This pattern, however, does not affect all cells equally. Only cells located between the phloem and pith are stimulated to undergo xylogenesis, whereas cells located between the phloem and epidermis are unresponsive. Whereas guard cell morphogenesis requires coordinated differentiation of two cells, morphogenesis of vessels requires coordination of thousands of cells. As each new vessel element differentiates from a young subapical parenchyma cell, it must recruit the next cell above itself to also develop into a vessel element, thus extending the vessel upward. And each element must make perforations aligned with the perforation of the element below it and the one above it. Each newly recruited cell must then recruit the next cell to be part of the vessel, and so on. Differentiation of sieve tubes requires not only the progressive recruitment of sieve tube members upward in the shoot's newly developing vascular bundle, but also the coordinated differentiation of a companion cell for each sieve tube member. In most cases, we assume that cells inhibit or promote activities in nearby cells by secreting chemicals, but it may be that the enlargement of some cells, or the lack of enlargement, causes physical stresses in tissues. Compression or stretching may also be important in establishing patterns of cells and tissues. Development and morphogenesis also occur at the level of organs and the whole plant. While still small, a seed plant embryo establishes a shoot apical meristem and a root apical meristem. The shoot apical meristem generates new cells for both itself and for the elongating stem. It also establishes leaf primordia in characteristic phylotactic patterns, and leaf primordia in turn establish axillary buds. Although each axillary bud develops its own apical meristem, a chemical messenger from the stem's apical meristem inhibits most of the stem's axillary buds, keeping them quiescent and thus controlling the pattern of branching and the shape of the plant. Vascular bundles differentiate upward toward the shoot apex and branch at the proper place such that leaf and bud traces interconnect the stem's vascular system with those of leaves, flowers, and branches. Figure 14, 1A cell has become a guard cell mother cell and is exporting the inhibiting peptide EPFL. The peptide enters adjacent cells and reacts with a protein produced by another gene, erecta. The combination of peptide and protein inhibits these cells from becoming guard cell mother cells, but cells located farther away receive no peptide and thus are not inhibited. For are shown as guard cell mother cells that are now inhibiting cells adjacent to themselves. Rarely, inhibition fails and the proper pattern does not develop. Here, too many cells develop into guard cells. This mutant gene form has been named too many mouths. GC equals guard cell. GCMC equals guard cell mother cell. Orange equals developing stomatal pore. Figure 14, 2A seed has just germinated, and the microscopic shoot apical meristem is probably just starting to make its first leaf primordia and the first cells of the nodes and internodes. At this point, it is impossible to tell what species this is as many Eudicot seedlings look just like this. But as new cells are formed, each differentiates in a particular way, guided by its genes and hormones such that the leaves have characteristic features, as do the nodes, internodes, axillary buds, and later on the flowers and fruits. Ultimately, the actions of all the genes guide the development of a plant body that is easy to recognize as being a particular species. This is the result of differential gene expression, as explained in this chapter. Plant bodies tend to be much simpler than those of animals, but even so, plant growth and development are not trivial. A great deal of information is required to establish polarity and symmetry, then patterns of cells, tissues, and organs. And the differentiation and morphogenesis that converts each pattern into actual cells, tissues, and organs require even more information. Virtually all information needed for plant development is stored as genes located in nuclei, plastids, and mitochondria. As an embryo develops and later as a seed germinates, it develops into a complete plant relying entirely on its own self-contained information and the intercommunication between its parts. But plants live in habitats with environmental factors such as light, gravity, water, temperature, seasons, and other organisms. All plants have evolved to have the capacity to detect at least some environmental factors and to alter their development and morphogenesis in adaptive ways. This is an important concept. Plants respond to certain environmental cues in ways that make the plants more likely to survive and reproduce successfully in the habitats they find themselves in. For example, all living stem and leaf epidermis cells synthesize some cuticle. If they are alive, they make cutenin. But many plants also can detect environmental moisture and respond adaptively by producing a thicker, more effective cuticle if conditions are dry and a thinner, less expensive cuticle if it is humid. Similarly, seeds germinate and grow if given water and warm temperatures, but if buried under several centimeters of soil when they germinate, they detect gravity and respond adaptively. Shoots grow upward, eventually reaching light, whereas roots grow downward into deep soil that may be more moist. Gravity is not necessary for growth, but it acts as a source of information about where the seedling is located with respect to light and moist soil. In these and many other cases, the environment provides information that allows plants to alter aspects of development and morphogenesis that would occur anyway. In other cases, environmental cues are needed for the plant to alter its development radically and to complete its life cycle. Many plants detect season by measuring the length of nights, and this stimulates the adaptive response of producing flowers at the right time of year or restricting growth and initiating preparations for winter. Without this environmental information, the plants would continue to grow vegetatively without ever forming flowers or without producing cold-resistant dormant buds, and this would be disastrous for the plant. Note that here too all information needed to actually produce flowers or cold resistance is present in the plant's genes, it is just that the plant itself cannot activate those genes until triggered by environmental signals. It is important to distinguish between adaptive responses and mere consequences of environmental stress. When fire kills a plant, the plant's death is a result of overly adverse conditions, it is not adaptive to the plant. Similarly, a late frost in spring may cause developing leaves to become malformed. The resulting shape is not adaptive, it is merely that frost disturbed leaf morphogenesis destructively. This chapter describes several environmental cues that plants detect, the chemical signals used by plant parts to communicate with each other, and several widespread types of development and morphogenesis. The following chapter discusses genes and the mechanisms by which their information is accessed and used. Environmental complexity. If all plants were extremely small and lived in completely uniform, non-varying environments, most would probably be simple and would experience little selective pressure for the evolution of complex shapes, tissues, organs, and metabolism. The most uniform, constant conditions occur in oceans and large lakes, where water buffers rapid changes in temperature, acidity, oxygen concentration, and other factors. Under such stable conditions, small organisms such as algae, protozoans, and sponges are simple. But most organisms exist in a heterogeneous environment. Gravity comes from only one direction. The sun is either to the side or overhead, but never below. Temperatures are lower on the shady side of a plant. Moisture depends on depth below or height above the soil surface. This mosaic of conditions changes over minutes, days, seasons, or longer periods of time. It is selectively advantageous for plants to sense these differences and changes and to respond to them. Most plants are so large that their bodies exist in several different microenvironments. Consider a small tree, its roots are in soil, which is usually moister, cooler, and darker than air. The highest branches are in open air, exposed to full sunlight and the full force of wind, storms, rain, and snow. The trunk base and lower branches are in an intermediate environment, less stable than soil and less variable and severe than open air. A vertical tree trunk is oriented to best resist gravitational attraction, whereas horizontal branches are highly stressed unilaterally by gravity. In springtime, the shoot can become warm enough for active metabolism even though the soil remains cold or frozen. The plant parts must communicate with each other, or shoot buds would become active and expand before roots were capable of transporting water to new leaves. In autumn, increasing night length and declining air temperature signal impending winter and the need for dormancy. Roots are informed about changing seasons by chemical signals from the shoot. The need for intercommunication and coordination also exists within a limited region of the body. For example, leaf parts act in a coordinated fashion during development such that the petiole has enough xylem and phloem to facilitate the transport needs of the blade. Too little conductive tissue causes the blade to suffer water stress or an inability to export sugars. Too much conductive tissue is a waste of energy and material. Even on the intracellular level, organelles must communicate with each other because their metabolisms are interrelated. All levels of communication have in common a basic mechanism. Information about the environment or the metabolic status of the organ must be perceived. The plant must sense environmental cues such as changes in temperature, moisture, or day length, or the nucleus must receive chemical signals if conditions in the surrounding cytoplasm change. Next, information must be transduced, or changed to a form that can be either acted upon or transported. Finally, there must be a response. The plant must enter dormancy, produce flowers, change the type of leaf production, and so on. If any of these steps is missing, the plant cannot respond to the environment. These principles apply to all organisms, and of course, higher animals have the most elaborate mechanisms. Their sophisticated sense organs for sight, taste, touch, hearing, and smell perceive external conditions. These sense organs transduce the perceived information to a transportable form such as nerve impulses or hormones that are secreted into the bloodstream. Animal responses are also typically highly elaborate, involving precisely controlled movement and increased activity of organs. Sensory systems and response mechanisms of plants tend to be simpler, and their signal transport is usually slow, involving movement through cortex parenchyma or phloem. This does not mean that animal systems are superior to those of plants. Superior has no meaning. The proper question is this: which is more adaptive, which is more advantageous selectively? In terms of evolution and natural selection, animal systems are successful adaptations for organisms that must detect food or predators and must move in order to capture that food or avoid being captured. Any animal with a plant-type mechanism of perception, transduction, and response would soon starve or be eaten. Figure 14.3 The signal for dormancy and preparation for winter is short days in autumn, perceived by leaves. The chemical messenger transported from the leaves causes the shoot tip to produce bud scales instead of leaves. The vascular cambium fills with many small vacuoles and stops mitosis and cytokinesis. Young xylem mother cells differentiate to a predetermined stopping point and then become dormant. Roots slow their growth greatly but usually do not stop completely. Roots and the cambial region cannot perceive the approach of winter themselves, but depend on leaves as sites of perception. Although the shoot apices could perceive it themselves, the entire plant is integrated as a whole by being cued by the leaves. On the other hand, rapid response mechanisms would not be adaptive in plants. They are extremely expensive to build and maintain and are unnecessary because most environmental conditions important to plants change only slowly. Eyes, nerves, muscles, or adrenal glands are not needed by plants for absorption of carbon dioxide, water, minerals, or light, or for perception of autumn and preparation for winter dormancy. For sexual reproduction, animal-pollinated plants do need sophisticated perception and response mechanisms, but basically, they simply rent those of their pollinators, paying with nectar or other rewards. Finally, the very sophistication of animal sensory response systems makes animals more vulnerable, more easily injured, and more dependent on avoiding dangers such as fires, floods, freezes, and predators. Plants are typically much more resilient than animals, being able to survive burning because of thick bark or resprouting from rhizomes and bulbs. Many withstand flood by being tough or flexible. Predators can consume most of a plant's leaves, wood, or roots without actually killing the plant. Light. Besides energy for photosynthesis, light also provides two important types of information about the environment: the direction or, more precisely, the gradient of light. This allows a plant to grow or to orient its leaves toward a region of bright light, which increases the light available for photosynthesis. The duration of light provides information about the time of year. Air temperature is unsuitable because cool autumn temperatures may be followed so quickly by severe cold that plants do not have enough time to become dormant. But day length is an infallible indicator of season. Gravity. It is selectively advantageous for many plants to orient themselves or their parts with respect to the direction of gravity. In some cases, gravity itself is important because it causes weight stress. A vertical stem supports more weight than does a similar stem growing at random. Whenever a plant is bent or tilted because of flooding or the slipping of a hillside, the plant must change its growth back to an upright direction. If it continued to grow at an angle, it would need to produce many more fibers to support its weight. In some situations, the direction of gravity is instead a guide to other important factors. Roots that grow downward are more likely to encounter water and minerals. Shoots that grow upward grow above other plants and encounter better conditions for photosynthesis, pollination, and seed distribution. Normally, shoots do this by growing toward the brightest light, the open sky, but shoots of seeds that germinate deep in the soil must determine which way is up while in the dark. Direction of gravity is their only reliable guide. Most bilaterally symmetrical flowers must be aligned with the body symmetry and flight pattern of their pollinator. Such flowers must be bilaterally symmetrical vertically and project horizontally because insects and birds do not fly upside down or sideways. The flower must orient itself along the same environmental gradient that the pollinator uses, the gravitational gradient of up versus down. Figure 14. 4 of these penstemon flowers have detected and responded to gravity in two ways. First, they all project horizontally, not at random and not toward the sun. Second, they are bilaterally symmetrical, and the plane of symmetry is vertical. Thus, flower symmetry matches the symmetry of bees, and flower projection allows bees to land easily. Weight is a source of information to the plant about the amount of parenchyma or sclerenchyma needed to counteract the gravitational attraction on the plant. Initially, the flower stalk was strong enough only to support the weight of a flower. By sensing and responding to gravity, it is now strong enough to support the weight of an apple. Although gravity does not change with time, the force it exerts on a particular organ does change as the weight supported by the organ changes. The pedicel of an apple flower supports almost no weight, but the same pedicel must later support the weight of a fully grown fruit. The extra fibers are not produced until needed. Similarly, a young branch must be strong enough to support a small amount of weight, whereas a larger branch must support more. Touch. Although plants do not move around like animals, their parts frequently grow against objects and respond to this contact. Certain types of contact are detrimental, for example, when a root grows against a stone or a branch rubs against another branch. In these cases, a thick bark is adaptive as a protective layer, and it is produced only where needed, only in the area being touched. Other types of contact are beneficial. After a tendril touches an object, it grows around the object and uses it as a support. When a fly touches sensitive trigger hairs on a Venus flytrap, the trap closes, catching and holding the insect during digestion. In some cases, the contact is between two growing primordia and is a normal developmental feature. Many flowers that have fused petals or carpels start with separate primordia that grow together and fuse, acting as a single unit during development. In each case, the physical action of touching is similar, but each organ responds in a distinct way that is adaptive for the plant. The response of each would be inappropriate if it occurred in the others. Temperature. Temperature fluctuates in a predictable pattern on both a daily and a yearly basis. Changing temperatures can induce many specific types of plant development. Although most plants appear to be quiescent and virtually lifeless in winter, a considerable amount of critically important metabolism is occurring. This metabolism usually does not proceed at temperatures above 1 degree Celsius to 7 degrees Celsius. Cold temperatures are required for the normal flowering of biennial and many perennial plants. Species of perennial trees that are adapted to habitats with cold winters, such as apples, typically require near-freezing temperatures to break the dormancy of their flower buds, which they had formed in the previous summer. If the trees are grown in areas with warm winters, they form flowers that never open. In contrast, biennial plants spend their first year in a vegetative phase and cannot be induced to form flowers. Their first cold winter vernalizes them. It causes them to switch to a state in which they can sense and respond to a stimulus that induces flower formation. In the year after the vernalizing winter, the plants resume vegetative growth, but when exposed to the proper stimulus, they respond by producing flower buds. In their first year, summer has no effect. In their second year, it induces flowering. If never vernalized, they never produce flowers. Low temperatures are required to induce deep dormancy in temperate trees and shrubs. Short days of autumn induce plants to initiate preparations for winter and enter a mild state of dormancy, but the most resistant stages are not entered until the plant actually experiences a week or two of cool temperatures. In contrast, cool temperatures are required to break the dormancy of many seeds, allowing them to germinate when their habitat becomes warm and moist. Figure 14. 5. The carpels of Catharanthus roseus are initiated separately and consist of protoderm and ground meristem, but they soon crowd into each other and fuse into one syncarpellus gynoecium. At the points of contact, the protoderm changes into mesophyll rather than epidermis. Figure 14.6. In its first year, a biennial plant has only a very short stem, with all its leaves attached close together, and is unable to flower. In its second year, a biennial plant produces a large group of flowers, almost always on a tall stalk. After the fruits and seeds mature, the entire plant dies. This is mullein, Verbascum thapsus. Water. Although water is an absolute prerequisite for life, its presence probably does not act like a signal in the way other factors do. If enough water is available, plants grow; if not, plants wilt and perhaps even die. Although roots often appear to grow toward water, they actually grow in all directions, and those which, by chance, grow toward water grow more rapidly because they are in a favorable environment. Roots that grow away from water grow slowly, but only because they enter an environment too dry to permit growth. Roots do not turn and grow toward water in the way they turn and grow toward gravity. Water scarcity triggers specific adaptive responses. One of the first is production of the hormone abscisic acid, which causes guard cells to lose potassium and close stomatal pores. This occurs in most plants even while cells have enough water to carry on basic metabolism. If water stress continues or becomes more severe, new responses are triggered that may inhibit production of new leaves, increase the cuticle on existing leaves, or even initiate abscission of leaves. Pests and damage. Plants must detect when they are being attacked by pests or being damaged physically. Pests such as bacteria, fungi, and insects attack plants chemically with toxins and digestive enzymes. Plants also suffer physical damage when larger animals trample them or chew into leaves and twigs. Plants have at least two mechanisms that perceive such damage. First, purely physical damage breaks a plant's own cell wall molecules, producing fragments of cellulose and hemicellulose. Although cell walls might be physically broken in many ways, most fragments will have certain physical and chemical patterns, such as bits of cellulose molecules protruding from the edges with portions of hemicellulose attached. These are called damage-associated molecular patterns, and when the fragments diffuse inward to a cell's plasma membrane, detector proteins bind to them because the proteins have shapes that match the bits and pieces of cellulose and hemicellulose. Such proteins are called pattern recognition receptor proteins. Once a pattern recognition receptor protein binds to a cell wall fragment, it initiates a signal inside the cytoplasm, informing the cell that its wall has been damaged. Second, pathogenic fungi and bacteria attack plants with digestive enzymes such that the microbial cells can penetrate the cell wall. Such an attack often produces damage-associated molecular patterns that alert the cell, but just as importantly, the microbes have their own chemicals that a plant can perceive. Fungus cell walls contain chitin, and fragments of chitin have distinctive patterns known as microbe-associated molecular patterns. The plant cell's plasma membrane has pattern recognition receptor proteins whose shapes match that of chitin, and which inform the plant a fungus is attacking whenever the protein binds a chitin fragment. Fragments of chitin are small, so they diffuse toward the plant cell's plasma membrane more rapidly than the fungus can grow there. Similarly, many plants have pattern recognition receptor proteins that recognize bits of bacterial flagella. The evolution of pattern recognition receptor proteins as part of a plant's self-defense mechanisms has been possible because of the ancient origins of chitin, flagella, cellulose, and hemicellulose. These have been reliable indicators of trouble that have not changed for hundreds of millions of years. Responding to environmental stimuli. Plant responses to the diverse types of information present in the environment can be grouped into four simple classes. Tropic responses. A tropic response is a growth response oriented with regard to the stimulus. For example, growth toward a bright light is a phototropic response. A positive tropism is growth toward the stimulus, a negative one is growth away from the stimulus, and plagiotropism is growth at an angle. Most taproots are positively gravitropic, growing downward in response to gravity, whereas shoots are negatively gravitropic, growing upward in response to gravity. Branches and secondary roots grow horizontally or at an angle, plagiogravitropically. When touch is the stimulus, the response is thigmotropism. Positive thigmotropism occurs when a tendril touches an object and, by growing toward it, wraps around it. P tendrils are extremely sensitive to touch. After a brief rubbing with a lightweight thread, tendrils gradually coil toward the stimulated side, even if a physical object is no longer present. Pollen tubes of flowering plants are suspected of displaying positive chemotropism, growing along the style to the ovary by following a gradient of chemical released from the ovule, probably from the synergids. However, this is still uncertain, the chemical responsible is not known, and pollen tubes would have to be sensitive to extremely slight variations in its concentration. Tropic responses often involve a change in direction of growth. Tendrils grow in a slow spiral, called circumnutation, until they touch an object, and then they change to oriented growth. Changing direction involves differential growth. One side of the tendril must grow more than the other. This can occur by growth on the contact side slowing, growth on the opposite side accelerating, or both. After a tendril starts differential growth, it continues growing differentially until it is mature a few hours or days later. In contrast, if a plant is tilted, its shoots begin differential growth until they are again oriented properly, and then they grow straight ahead with equal growth on all sided, perhaps for years. Table 14. 1 Types of plant responses to stimuli. Figure 14, 7 Construction of porch steps trap these plants, but by growing toward light, their shoot tips found spaces between boards and emerged from a suboptimal dark environment into a more suitable sunny environment. Sunflowers have an unusual type of phototropism. Their flower follows the sun all day long, so they turn from east to west every day for several weeks until finally the stalk becomes too tough and woody to move. Notice that every sunflower is facing the same direction, almost directly toward the sun. Figure 14.8 The central shoot of this tree grows vertically, probably due to both positive phototropism and negative gravitropism, but the branches grow at an angle to vertical, plagiotropically. Table 14. 2 Prefixes for stimuli. Gnostic responses. A gnostic response is a stereotyped non-growth response that is not oriented with regard to the stimulus. For example, the trap leaf of a Venus flytrap has 6 large, sensitive trichomes. If a fly or other insect touches any 2 of these, the trap closes. It does not matter if the fly was moving north or south or up or down, the trap always closes in the same manner in this thigmonastic response. Furthermore, the trap does not grow shut, it closes as motor cells on the midrib upper side suddenly lose turgor. Positive and negative are not used because the response is not oriented with regard to the stimulus. Many pollinators are active only at night or during the day, and the flowers they pollinate are open only at the appropriate time. Sepals and petals spread open when the sun rises in diurnal species and as it sets in nocturnal ones. Although presence of the pollinator is the critical factor for pollination, the cue that stimulates flower opening is the presence or absence of light, not the presence or absence of pollinators. The opening and closing always happen in the same manner, even if light is given artificially from the west, north, or south, above, or below. This response is photognostic. Gnostic movements are based on changes of turgor pressure rather than growth, thus, movements can be repeated. This occurs in many leaves that undergo sleep movements with the blade elevated in the day, lowered at night. Such leaves often have an enlarged area, a pulvinus at their petiole base, and it contains motor cells that cycle between being turgid and flaccid. Morphogenic responses. A morphogenic response, sometimes called a morphogenetic response, causes a change in the quality of the plant, that is, a fundamental change occurs in the metabolism of a tissue or even the entire plant. Because day length is such an excellent indicator of season, photomorphogenic responses are numerous: the induction to form flowers, the induction of dormant seeds to germinate, and the induction of buds to become dormant. An example of a gravomorphogenic response is the formation of fibrous wood when a stem or branch is tilted and becomes stressed by gravity. Thigmomorphogenic responses include formation of extra bark where branches rub against an object and formation of a suture when petal or carpal primordia grow against each other. Figure 14.9 Conversion from the vegetative to the floral condition is a common photomorphogenic response. Day length is controlled in these commercial greenhouses to ensure that all the poinsettias bloom simultaneously at Christmas. The growers could just as easily make them bloom on the 4th of July by controlling day and night length. Figure 14:10 in these seeds and many others, a two-part mechanism ends dormancy, cold winter temperatures, and rain destroy or wash out an inhibitory chemical, and then light triggers germination. The light-dependent mechanism ensures that the seeds do not germinate while deeply buried under leaf litter and soil. Red light induces germination, but far-red light blocks germination. Taxis. Taxis is a response in which a cell swims toward or away from a stimulus. Even in plants like mosses, ferns, cycads, and maidenhair tree, sperm cells swim to egg cells by following a chemical gradient. In algae, chemotaxis is similarly important for reproduction, and in many species, phototaxis allows them to swim toward light for photosynthesis or away from light that is too intense. Communication within the plant. Perception and transduction. Many, possibly most, responses occur in tissues or organs different from those that sense the stimuli. The site of perception is not the site of response, so a form of communication must exist. In plants, most sites of perception and response are not specialized for those functions, but seem to be rather ordinary cells. Day length is probably perceived by all living leaf cells. No specialized region of cells has been discovered. Low temperatures for vernalization appear to be detected by buds, which do not contain a particular group of cells specialized just for temperature perception. In root caps, certain cells called statocytes do have large starch granules, statoliths, that sink in response to gravity. Statoliths are too dense to float in cytoplasm and always settle to the bottom of the cell, thereby distinguishing down from up. This is our best example of a set of specialized perceptive cells. The trigger hairs on Venus flytrap leaves are also a discrete perceptive mechanism, but it is not known which cells within the hairs are responsible. The site of perception is tentatively assumed to be the site of transduction, where the stimulus is converted into a form that can be transmitted and can trigger a reaction at a response site. Transduction is still a complete mystery in almost all plant responses. We do not know how changes in temperature, light, weight, or humidity are converted into chemical signals. Two factors are important in perception and transduction: presentation time and threshold. Presentation time is the length of time the stimulus must be present for the perceptive cells to react and complete transduction. Presentation time for root. Gravitropism is easy to understand. A root must lie on its side long enough for statoliths to sink to the new bottom of the cell. If the root is returned to vertical before they can settle, no perception occurs. In many tropic responses, only a brief touch or unilateral lighting is sufficient to cause curvature. Presentation times are often only a few seconds. After the stimulus has acted long enough to fulfill the presentation time, a response occurs even if the stimulus is removed. For example, the vernalization of many biennial plants has a presentation time of only 1 or a few days. After this, the plants still flower at the proper time even if kept in warm, non-vernalizing conditions. Tendrils of peas do not bend thigmotropically in the dark, but if they are rubbed for several seconds, their presentation time, in the dark, they bend when placed in light even though they are no longer being touched. Figure 14:11 cells located centrally in this root cap are statocytes, and their starch grains are statoliths. Regardless of vertical or horizontal position, the statoliths are located at the gravitationally lower side. It is necessary to distinguish between the gravitational and the morphological bottom in gravity-sensing systems. Threshold refers to the level of stimulus that must be present during the presentation time to cause perception and transduction. In phototropism, plants are extremely sensitive to very dim unilateral light. If they are in an otherwise dark environment, the threshold for curvature is low. In bright conditions, the threshold is higher, and the unilateral light must be much stronger to trigger curvature. In Venus flytraps, the threshold for stimulating leaf closure is moderate. The trigger hairs must be firmly bent. This is advantageous in preventing wind or rain from triggering trap closure. The moderate threshold almost guarantees that the trap contains an insect every time it closes. Figure 14, 12 threshold must be appropriate to the amount of change a stimulus can cause. If Venus flytrap hairs had an extremely low threshold, they would be capable of detecting and catching every insect, but wind and rain would cause so many useless closings that the leaf would be inefficient because it would miss insects whenever it was closed unnecessarily. With a medium threshold, it captures more insects because it is open and ready much of the time. Small insects may escape because they cannot bend the trigger hairs enough to meet the threshold. If the threshold were too high, no insects would be caught because none could bend the hairs. The trap would close only when larger animals brushed against it, but these animals are too big to be enclosed in the trap, thus, all closures would be unproductive. We can hypothesize that natural selection results in a threshold appropriate for the most abundant size of insects. Related to threshold is the level of response relative to the level of stimulation. The alternatives are all or none responses and dosage-dependent responses. In an all or none response, after the threshold and presentation time requirements are met, the stimulus is no longer important, the response is now completely internal. Individuals respond identically whether they receive strong or weak stimuli, regardless of whether the stimulus was brief or long-lasting. For example, many species are induced to flower by environmental conditions. After the minimal threshold and presentation time requirements are met, the plants flower fully, limited only by their general health, vigor, and nutrient reserves. Until they receive the proper stimulus, they produce no flowers. Their flowering is all or none. Examples are poinsettia, chrysanthemum, hibiscus syriacus, and oats. In dosage-dependent responses, the amount or duration of the stimulus affects the amount or duration of the response. In species of this type, individuals that receive only minimum stimulation flower poorly, even if the plant is quite healthy. Those that receive longer or stronger stimulation produce more flowers. Examples are turnip, marijuana, and some varieties of cotton and potato. Chemical messengers. Almost all plant communication is by a slow mechanism, transport of hormones through the plant. Hormones are organic chemicals produced in one part of a plant and then transported to other parts, where they initiate a response. A critical aspect is that hormones act at very low concentrations. Hormones are synthesized or stored in regions of transduction and are released for transport through either phloem or mesophyll and cortex cells when the appropriate stimulus occurs. At the site of response, hormones bind to receptor molecules, usually located in the plasma membrane, and thereby trigger a response. Hormones appear to be released into general circulation and are not carried specifically to the target. Many regions that are not target regions are exposed to the hormone but do not respond because they do not have the proper receptor molecules. In some instances, a plant hormone acts directly on the cells that produce it. At one time, plant hormones were believed to carry in their structure much of the information necessary for the response. We now know that plant hormones are quite simple in structure. The receptor cell and its nucleus contain almost all of the information necessary for proper response, and hormones serve only to activate the response. An analogy is a computer, its programs, and commands. The computer is capable of carrying out numerous functions and processes, but only if properly controlled. The same is true of cells, tissues, organs, and entire plants. Computer programs contain the information needed to run the computer, just as the nuclear, plastid, and mitochondrial genes contain the information needed to run cells. Both computer programs and genomes contain numerous sets of information. On a computer, commands such as open, print, save, and copy select subroutines or programs. Hormones are thought to act as commands that activate programs within the target cells. In higher animals, because so many systems and responses must be activated, many distinct hormones are necessary. Because plants are much simpler, their responses can be controlled by fewer hormones. Even so, the handful of known plant hormones seems inadequate. It is likely that many plant hormones are still unknown to us. Also, many responses are activated not by one hormone but by a combination or a sequence of several hormones, or a particular hormone elicits different responses when present at different concentrations. The following are examples of the most well-studied hormones. Figure 14.13. A cell responds to a hormone only if it has receptors for that hormone. Evidence suggests that some receptors are in the plasma membrane and that others are in membranes such as the endoplasmic reticulum. Once bound, the hormone receptor complex may cause a metabolic change immediately, or the complex may migrate to another site, such as the nucleus. In many responses, some nuclear genes are activated and others are repressed. Cells may have receptors for several hormones. If hormone A is present, it binds and activates program A. Other programs are unchanged. One of the results of program A might be to withdraw the receptors of either A or B from the membrane, or to add receptors for C or D, thus changing the sensitivity and type of response possible. A second cell may have a different program activated by hormone A. The response is cell-specific, not just hormone-specific. The effects of a hormone are often quite different when the hormone is applied alone or with or after a second hormone. In this hypothetical cell, development occurs only if hormones A and B are applied simultaneously, or if hormone D is applied after hormone C. Auxins. The first plant hormone discovered was auxin. In 1926, it was identified as the chemical messenger involved in positive phototropism in oat seedlings. Identifying it chemically at that time was impossible because it is present in such low concentrations. Experiments had to be performed by allowing auxin to diffuse out of a seedling leaf tip into a small block of agar, which was then used as if it were a small dose of auxin. See figures 14.25g and 14.25h. The auxin was later identified as being indolacetic acid, which could be synthesized artificially and applied to plants under various conditions to find other responses that IA might either mediate or inhibit. The search was successful. Dozens of responses were found. Many compounds chemically related to IA were also found to be as effective or even more so. It was hypothesized that IA was only one of many natural auxins, each with its own effect and role, but further findings were not consistent with that hypothesis. Analysis of IA metabolism showed that the compounds were converted to IA by the plant's enzymes. Many synthetic compounds mimic the effect of auxin or the other hormones. For clarity, only natural products are called hormones. The term plant growth substance is used for any hormone-like compound, natural or artificial. Naphthalene-acetic acid, an artificial compound, produces effects in plants that are for the most part indistinguishable from those of IA. 2. For dichlorophenoxyacetic acid is auxin-like but so powerful that it disrupts most normal growth and development even in low concentrations, making it valuable as an herbicide. Figure 14.14 IA is the natural hormone auxin, the others are synthetic. They are called plant growth substances. IA is related to the amino acid tryptophan. So far, at least 4 separate metabolic pathways are known that convert tryptophan to IA. Some plants, corn, for example, synthesize IA by different pathways at different stages of development, and each pathway has its own characteristic set of controls. The most active centers of auxin synthesis are shoot apical meristems, young leaves, and fruits. IA is present in root tips, but is believed to be transported there from the shoot rather than being synthesized there. The concentration of substances as powerful as hormones can be controlled not only by synthesis but also by destruction and by conversion to an inert storage form. 2 pathways for IA destruction are known: removal of the side group and oxidation of the 5-member ring. IA is converted to an inactive form by conjugating it to various compounds. In the conjugated form, IA is safe from destruction, it can be stored indefinitely in seeds, and it can be transported from cotyledons to the epicotyl during germination. Conjugation allows rapid regulation of the level of free IA. Up to 80% of the IA in oat seeds is conjugated, but this can be deconjugated, releasing free IA during germination more quickly than synthesis could. Table 14. 3 examples of responses involving hormones. In addition to hormone transport by phloem, a second mechanism exists for auxin-only, polar transport. In shoots and leaves, auxin moves bacipitally, from the apex to the base of the plant, and in roots it moves acropitally toward the root apex. Movement is about 11 mm hr regardless of whether the tissue is in a vertical, horizontal, or upside-down orientation. By means of the polar transport system, auxin movement through the plant can be maintained independently of the variation in phloem transport caused by changing sinks and sources for carbohydrates and minerals. Cytokinins. Cytokinins were named for the fact that their addition to a tissue culture medium containing auxin and sugar stimulates cell division, cytokinesis. The first one discovered, kinetin, is an artificial cytokinin. However, two natural ones, zeatin and isopentenyl adenine, have been found, and more are suspected to exist. Cytokinins are purines related to adenine. Extensive testing of adenine analogues has been done to determine which aspects of its chemical structure are critical to the molecule's ability to act as a cytokinin. The most active compounds have a side group containing 4 to 6 carbon atoms attached to C. If this side group is longer or if complex groups occur at other areas, the molecule does not act like a cytokinin, apparently lacking the proper shape and charge to bind with the cytokinin receptor molecule. Figure 14, 15 cells of most dicots, such as this tobacco, can be grown in culture if provided with auxin, cytokinin, some vitamins, minerals, and sugar. The ratio of auxin to cytokinin is important, as are the absolute concentrations. At 1 ratio, the cells proliferate as a callus composed of parenchyma. At another ratio of auxin to cytokinin, buds form in the callus and then grow into embryos. Figure 14, 16 Both natural and artificial cytokinins are related chemically to adenine. The size and chemical nature of the group on C is critical. Like auxin, cytokinins are involved in dozens of responses in all parts of the plant. One important response is root-shoot coordination. As roots begin to grow actively in the spring, they produce large amounts of cytokinins that are transported to the shoot, where they cause dormant buds to become active and expand. The richest concentrations of cytokinins generally occur in endosperm and are apparently involved in controlling the development and morphogenesis of the embryo and seed. Gibberellins. At least 125 gibberellins are known, and rather than being named, they are just numbered, GA125. GA has the name gibberellic acid, and at first it received the greatest amount of study because it could be obtained easily from fungi and used in experiments. However, it now appears that GA and GA19 are more active and important in plants. Gibberellins have diverse functions but a unifying structure, the gibberellin ring system. This class of hormones is thus defined by structure. A compound cannot be a gibberellin if it does not have the gibberellin ring system. Figure 14, 17 All gibberellins are based on gibberellin. The most common forms are GA and GA. Not all of the 125 gibberellins occur in plants, at least 15 have been found only in fungi. Gibberellin metabolism is complex. Only a few gibberellins are known to be active as hormones, others are precursors or intermediates in transforming one active form into another. Relative concentrations of the various gibberellins change in response to environmental signals. When spinach is exposed to long days, the level of GA19 undergoes a 5-fold decrease, GA20 and GA29 increase drastically, but GA17 and GA44 do not change. As a result of these changes, spinach stems begin to elongate. Long days are the stimulus, shoot elongation is the response, and alterations of gibberellin levels is the mechanism linking the two. Gibberellin metabolism appears to occur in all parts of the plant, but seeds, roots, and leaves are especially important. Gibberellin transport is studied by giving plants synthetic, radioactive gibberellins and then tracking the movement of the radioactivity. Both xylem and phloem sap become radioactive, and it is believed that the large amounts of gibberellins synthesized by active root tips are loaded into xylem. Gibberellin movement from leaves and seeds appears to correlate more closely to phloem sap dynamics. Like all other hormones, gibberellins are involved in numerous responses. Abscisic acid. This class apparently contains the single compound abscisic acid. As its name suggests, it was thought to play a role in the abscission of fruits, leaves, and flowers, but currently, we do not know whether that is true of many species or only sycamore, the species in which it was discovered. ABA is widely regarded as a growth inhibitor, possibly involved in inducing dormancy in buds and seeds. However, dormancy is not just inhibition of growth, but a complicated set of changes that prepare the plant or seed for adverse conditions. ABA appears to be especially involved in many types of stress resistance. Heating leaves, waterlogging roots, chilling, and high salinity have all been found to cause sudden increases in ABA. If healthy plants are pretreated with ABA, they become much more resistant to stressful conditions. When plants begin to wilt, the concentration of ABA in leaf cells increases dramatically from about 20 μg/kg fresh weight to 500 μg/kg, and guard cells close stomatal pores. Wilt-induced production of ABA overrides all other stomatal controls. Other mechanisms that normally cause stomata to open are ineffective when ABA is present. The stimulus for ABA production appears to be soil dryness, which is detected by roots. When soil dries to some critical level, ABA production is dramatically increased and the ABA moves upward through the xylem to the leaves, causing stomatal closure. ABA can be removed by being converted to phthalic acid, which has no known hormonal activity. Figure 14-18. Many biennial plants grow as a rosette in their first year and then elongate rapidly in their second year, producing a tall shoot that bears numerous flowers. The bolting is controlled by gibberellic acid. If it is absent, the plants remain short. The plant shown here, Arabidopsis thaliana, completes this cycle in just a few months, not 2 years. All its large leaves are arranged in a rosette at ground level. After it was several months old, it bolted and produced the flowering stalks with long internodes. Figure 14-19. ABA is transported rapidly between cells and through phloem, therefore, its presence in a tissue is not proof that it was produced there. ABA can be synthesized from mevalonic acid in roots, stems, leaves, fruits, and seeds of various species. Alternatives. Box 14-1. Simple bodies and simple development in algae. Angiosperms have complex bodies, which require them to control many aspects of development, but there are alternatives. Green algae have much simpler bodies. Unicellular algae, such as Chlamydomonas, have bodies consisting of just a single cell. No middle lamella is formed during cell division, therefore, nothing holds the two daughter cells together and they just swim away from each other. Each individual has a body but no tissues or organs. They develop polarity with a front end that faces forward as they swim and a rear end. Colonial green algae are slightly more complex. These cells also lack a middle lamella but instead produce a gelatinous material that holds cells near each other. As a zygote divides and multiplies, all daughter cells remain loosely bound together. There is so little interconnection and interdependency that each group is considered to be a colony of individuals rather than the body of a single individual. In Gonium, each colony contains only a few cells, and the only sign of organization is that all flagella beat in a coordinated fashion. Pandorina is slightly more derived because it shows a trace of differentiation. The colony swims in one direction, and anterior cells are slightly different from posterior ones. Volvox colonies contain up to 50,000 Chlamydomonas-like cells and are easily visible without a microscope. Slight differentiation exists in that up to 50 cells in the posterior half of a colony are specialized for reproduction only. In green algae that do produce a middle lamella, daughter cells remain bound together and form a single integrated individual with a multicellular body. After the zygote of Eulothrix divides, one of the two daughter cells becomes a colorless adhesive holdfast cell that glues itself to a stone or seashell. The other daughter cell produces no adhesive, and its plastids develop into chloroplasts. Already the body has polarity, differentiation of two cell types, and can detect which cell is in contact with a solid substrate. The chlorophyllous cell divides repeatedly but always with crosswalls parallel to each other, and thus, the body grows as a radially symmetrical filamentous body, one cell thick but many cells long. This requires morphogenetic mechanisms that precisely control the orientation of cell division. In some filamentous species, a few cells undergo a longitudinal division, and then one of the two daughter cells grows out as a filamentous branch. Laminar bodies, such as those of ulva, are slightly more complex. A cell settles down and begins to grow into a ulithrix-like filament, but its cells divide in two directions and thus form a sheet. Then all cells divide once in a third direction. As a result, the sheet becomes 2 layers thick. Both filamentous and laminar bodies remain simple with regard to cell differentiation, having only holdfast cells and chlorophyllous cells. Also, their bodies are soft and flexible, being supported and moved by water, so they do not have phototropism or gravitropism. Figure B14, 1a colonies of volvox, all cells are almost identical, a few are specialized for reproduction. Little differentiation occurs. Figure B14, 1b individuals of the laminar alga ulva are 2 cells thick. The holdfast is not visible here. Several groups of green algae have parenchymatous bodies. Chara has a stem-like body up to 1 m long, divided into nodes and internodes, with whorls of branches arising in a precise pattern at internodes. The body is several cells thick, with large multinucleate cells in the center of each internode and smaller uninucleate cells surrounding them as well as composing the nodes and branches. All constitute a true parenchyma tissue because they are derived from an apical meristem that contains a prominent apical cell that divides in all 3 planes. Reproduction in Chara is significant. Its sperm cells are produced in a truly multicellular structure whose outer cells are sterile, only the inner cells convert to sperm cells. At maturity, the outer cells separate slightly and the motile sperm cells swim away. The egg is formed as the terminal cell of a short filament 3 cells long, but the subterminal cell subdivides, and those cells grow upward and surround the egg, differentiating into a variety of specialized cells. After fertilization, the sterile cells surrounding the fertilized egg deposit thickenings on their inner walls, those adjacent to the zygote. The resting structure thus consists not only of a thick-walled zygote but also protective sterile cells. Both sperms and eggs are produced in gametangia, consisting of 2 types of tissue. Reproduction requires the integrated functioning of several types of cells. This is an organ level of differentiation. Furthermore, germination of the zygote is reported to be promoted by cold temperatures and by red light. Development responds to environmental cues. Although all algae are much simpler than true plants, all undergo at least some development and morphogenesis, and Chara and its relatives have numerous sophisticated developmental metabolisms. Figure B14, 1c bodies of Chara are several cells thick and are true parenchyma, formed by an apical meristem. Bright orange round structures are about to release sperm cells. Yellow, elongate structures contain eggs. Each of these gametangia consists of some fertile cells surrounded by several types of sterile cells. Individuals of Chara undergo considerable differentiation. Ethylene. Ethylene is the only gaseous plant hormone, and it has the simplest structure. Its most commonly studied effects occur during fruit development. Fruits such as apple, avocado, banana, mango, and tomato are climacteric fruits. They ripen slowly as they mature, but in the final stages, numerous developmental changes occur rapidly. Starches are converted to sugars. Cell walls break down and soften. Flavors and aromas develop, and color changes. Ethylene stimulates these changes, but at first, so little ethylene is present that the changes occur slowly. However, one effect of ethylene in these fruits is the production of more ethylene, which constitutes a positive feedback system. The concentration increases exponentially and rapidly. The sudden burst of ethylene and the rapid completion of maturation of the fruit are known as a climacteric at this time. Ethylene production can be as high as 320 nlghr. In non-climacteric fruits, such as cherry, lemon, and orange, ethylene does not stimulate its own production, therefore, ethylene levels remain stable and no sudden change occurs just before maturity. Because ethylene controls the ripening of most of our important food fruits, it is important commercially. It is used in harvesting cherries, cotton, and walnuts by causing their uniform abscission. It also synchronizes flowering and fruiting in pineapple, making harvesting easier. Ethylene can be drawn out of unripe fruits by storing or transporting them in a partial vacuum. When they reach market, air pressure is returned to normal, ethylene accumulates, and ripening occurs. Fruits may also be treated with 2-chloroethylphosphonic acid, which breaks down and releases ethylene. Figure 14.20 Ethylene is a simple, small molecule. Many of its effects are blocked by carbon dioxide, whose size and shape are similar enough to those of ethylene that it can bind to ethylene's receptor and block normal response. Bananas produce large amounts of ethylene as they ripen. This diffuses away and stimulates other types of fruits to ripen rapidly. People often place a single banana, or even just a banana peel, into a plastic bag with other fruits. The plastic traps the ethylene, which then causes the other fruits to ripen faster and more fully. Similarly, a bruised apple produces large amounts of ethylene, which can cause other apples to over-ripen. Thus, the old saying, "One bad apple spoils the barrel." Being a gas, ethylene moves rapidly through tissues by diffusion rather than by specific transport mechanisms. In many cases, it acts as a final effector for auxin. Arrival of auxin at a target site often causes that site to produce ethylene, which diffuses rapidly and triggers responses in the adjacent area more quickly than the auxin itself could. Being a gas also allows ethylene to escape from plants through stomata and lenticels, which makes it a means of detecting flooding. When rivers flood, ethylene builds up in submerged stems and induces the formation of parenchyma in certain species. Other hormones. Several other compounds show hormone-like activity but are either not present in all plants or are much less well studied. Brassinosteroids are complex chemicals involved in leaf morphogenesis, root and stem growth, and vascular differentiation. If plants of Arabidopsis thaliana are artificially mutated to disrupt brassinosteroid synthesis, the plants develop abnormally but can be rescued by applying small amounts of brassinosteroids. Jasmonic acid is involved in defense against animals and fungi. When animals chew into plants, membrane lipids are released, and one of them, linolenic acid, is converted to jasmonic acid. This travels to healthy cells and induces them to activate the genes necessary to synthesize alkaloids and chemicals that inhibit protein-digesting enzymes. Both of these interfere with an animal's ability to eat and digest. When fungi attack plants, they secrete small peptides and oligosaccharides, and several of these somehow cause jasmonic acid to be produced. It then stimulates plant cells to form an antifungus compound. Plants can attach a methyl group to jasmonic acid, creating methyl jasmonate, which diffuses away from the plant and stimulates adjacent plants to become resistant to pathogens. Salicylic acid, related to aspirin, is involved in resistance to pathogens, especially viruses. Artificial inoculation of leaves with viruses causes salicylic acid levels to rise, and these in turn activate disease resistance genes. The resistance mediated by salicylic acid is not confined to just the site where the viruses were applied. Instead, all parts of the plant become resistant. This is known as systemic acquired resistance. As with methyl jasmonate, methyl salicylate is volatile and is carried by wind to other plants, alerting them to the presence of pathogens in the area. Both chemicals are part of a method of plants communicating with each other. Also, staminate flowers of voodoo lilies produce salicylic acid, which then travels downward through the thick inflorescence axis until it reaches the base. There it induces certain cells to undergo thermogenic respiration, heat up, and give off foul-smelling fragrances that attract pollinators. Signal amplification cascades. Most chemical messengers are not transported specifically from the tissues that produce them into the tissues that will be controlled by them. Instead, the cells that produce them release them to be moved symplastically or apoplastically or into phloem. Consequently, even though a source might provide quite a bit of hormone, the concentration decreases as the hormone spreads through the plant, and only a few hormone molecules might actually bind to receptor molecules on the target cell. The signal is very weak and must be amplified by the target cell. Most plants and animals are diploid, so most nuclei have only 2 copies of each gene. Imagine that a hormone needs to activate a particular gene, perhaps one for cell growth. Each diploid nucleus has approximately 60,000 genes and several billion nucleotides. the chances are very low that a few hormone receptor complexes would diffuse from the plasma membrane into the nucleus and somehow encounter the correct genes. It is more likely that some would break down in the cytoplasm or be absorbed by the vacuole or spend days going from one gene to another, never finding the correct two. In all cases that have been studied, these weak signals are amplified by the target cell. When a hormone binds to its receptor, the receptor usually changes shape and becomes an active enzyme, typically a kinase, an enzyme that transfers a phosphate from ADP onto some other molecule. Because the hormone receptor complex is an enzyme, it functions repeatedly. The single act of one hormone molecule binding to its receptor is amplified into hundreds of molecules being phosphorylated. What do the second, newly phosphorylated molecules do? Do they go into the nucleus and bind to DNA? No. Typically, they also become active kinases and thus phosphorylate thousands of a third type of molecule, amplifying the signal even further. At this point, there are now thousands of messenger molecules, not just the original 1 or 2 or 3, so chances are very high that many will enter the nucleus and some will encounter the proper genes quickly. This is called a signal amplification cascade. An example will make this clearer. One of the most important signal amplification cascades is the MAP kinase cascade. It plays an important role in signal amplification for many types of development in plants and animals, and is believed to have arisen very early in the evolution of eukaryotes. Hormone binding converts the receptor into a kinase called MAP kinase. This strange name means that there are 3 steps in this amplification cascade. MAP3K phosphorylates and activates hundreds of molecules of MAP kinase, each of which phosphorylates thousands of molecules of MAP kinase. MEPK then finds and activates various factors needed to control gene expression and development in the target cell. This cascade allows one hormone molecule to trigger the activation of thousands of important regulatory molecules within just a few seconds or minutes. It also turns out that MAP3K phosphorylates several factors in addition to MAP2K, and similarly MAP2K activates various other factors besides MEPK. At each step, more and more types of response factors are being activated. Many other signal amplification cascades exist, some triggered by certain hormones, others being found only in certain types of response. The various cascades interact in some situations. For example, 2 hormones might both activate the same cascade such that the 2 hormones reinforce each other's effect. In contrast, the effect of one cascade might be to shut down or modify another cascade such that one suppresses or alters another. Activation and inhibition of shoots by auxin. Auxin is often described as a growth hormone, whereas ABA is considered an inhibitor. Unfortunately, such characterizations are confusing. Hormones simply carry information about the status of a particular region, nothing more. Whether the elicited response is inhibition or activation depends on the site of response. An example of the complexity is provided by shoot tips. As shoot apical meristems grow and initiate the new cells of shoots and leaf primordia, they also produce the auxin, IA. Young leaves are also a rich source of this hormone. No external signal must be perceived to initiate auxin production. Instead, this is a means of integrating the plant during ordinary growth. Large quantities of auxin indicate to cells that shoots are elongating and producing new leaves. Although neither signal perception nor transduction occurs, transport takes place. In stems, auxin undergoes basipital, polar transport at a speed of 5 to 20 mm per hour, perhaps by means of molecular pumps in plasma membranes. This downward flow of auxin surrounds all stem cells, and at least 3 cell types are set to respond to it, each response unique to the particular cell type. Cell elongation. In cells of the young internodes just below the apical meristem, auxin triggers cell elongation. When IA contacts these responsive cells, which are prepared for growth, it binds to a receptor, thought to be a small protein called ABP1. The cells begin to transport protons actively out across the plasma membrane. This has the effect of acidifying the cell wall. The protons break some of the chemical bonds that hold one cellulose microfibril to another and activate enzymes that weaken other bonds so that the wall becomes weaker. If the protoplast is turgid and pressing against the wall, it exerts enough pressure to stretch the weakened wall and growth results. Immature cells neither excrete protons nor grow if auxin is lacking. At lower internodes, fully grown, mature cells apparently lack the proper auxin receptors because auxin does not cause them to extrude protons or grow. Figure 14:21 cells in the subapical region have auxin receptors. If the apex is growing and producing auxin, Ia is present to bind to the receptors. After stimulation by auxin binding, the plasma membrane pumps protons from the cytoplasm into the wall, weakening it and allowing turgor pressure to stretch it. Cell elongation stops once the maximum cell size is reached, and adding more auxin does not cause any more elongation. Perhaps the receptors have been removed from the membrane. Apical dominance. The second site of response to apically produced auxin is the buds located in leaf axils. Their response is not cell elongation but rather inhibition of growth. Apically produced auxin induces dormancy in these axillary buds, the result being that each shoot tip has only one active apical meristem, a phenomenon called apical dominance. This is a threshold response. As the terminal shoot apical meristem grows away, the concentration of auxin around an axillary bud gradually decreases until at some point it drops below the threshold. Inhibition cannot be maintained, and the axillary bud becomes active and grows out as a branch or flower. As the axillary bud grows, it produces auxin but does not inhibit itself, although it does inhibit all its own newly formed axillary buds. Differentiation of vascular tissues. The third site of response to auxin produced in shoot tips is the vascular cambium. The response is cell division and morphogenesis. In springtime, as air temperatures rise and buds become active, their auxin moves bacipitally, activating the dormant vascular cambium. Auxin not only stimulates cambial cells to begin mitosis and cytokinesis, but also causes new daughter cells to differentiate into xylem cells. If an apical meristem is destroyed, by insects or a late frost for example, the basipital flow of auxin stops, vascular differentiation is interrupted, internode elongation ceases, and apical dominance is broken. Some axillary buds, now free of apical dominance, become active and re-establish a flow of auxin that maintains the vascular cambium and any other cells that depend on it. Three separate target tissues give three distinct responses, not because there are three separate chemical messengers, there is only auxin, but because part of their previous differentiation was preparation to respond to auxin in a particular way. It is important to realize that auxin carries no information except that the shoot apex is healthy and active. Each target site must have receptor molecules that interact with Ia and therefore detect its presence or absence. It is not known whether all three targets have the same receptors or whether each has a unique type, nor is it known how the interaction of Ia with the receptor triggers the response. Interactions of hormones in shoots. In some species, apical dominance may involve only the presence or absence of auxin. In others, there is an interplay of two or three hormones. Active roots synthesize cytokinins that are transported to the shoot and stimulate axillary buds. Whether buds become active or remain dormant depends on the relative amounts of the two hormones. If a plant is growing vigorously, its roots are active and cytokinin levels are high. Many buds at a distance from a shoot apical meristem have a low auxin-high cytokinin ratio and become active. Such a mechanism is adaptive because if a plant is growing well, activating more dormant buds increases the rate of new leaf production. The role of ABA in apical dominance is uncertain. It is present in quiescent buds, but does not decrease either just before or as buds are becoming active and growing out. Apical dominance in prickly pear cacti is more elaborate. The spine clusters are short shoots, and the spines are highly modified leaves. If the spine cluster is excised and placed in a tissue culture with cytokinin, the dormant short shoot apical meristem grows out as a long shoot, a new pad similar to a normal branch. If the culture medium contains gibberellins instead of cytokinin, the short shoot apical meristem produces more spines. It acts as a rejuvenated short shoot. In the last few years, we have made great progress in understanding the ways in which hormones and peptides interact to control shoot apical meristems. Cells in the meristem itself divide and grow, producing more cells for the shoot. The rate at which new cells are produced must match the rate at which some cells stop dividing and differentiate into mature cells of epidermis, cortex, xylem, and so on. If too few cells differentiate, the meristem would enlarge. If too many differentiate, the meristem would shrink. In the center of the shoot apical meristem, some cells express a gene named WUSCHL, which produces a protein that stimulates growth and division of meristem cells. If only WUS were active, the meristem would constantly grow larger. But another group of cells expresses the gene CLVATA3, which limits meristem size. The two make a negative feedback loop because each affects the other. WUS activates CLV3, which inhibits WUS. If WUS becomes too active, it stimulates CLV3, which then inhibits it. But if CLV3 is too active, WUS becomes too quiet and the cells are not stimulated to make CLV3. Other genes and their peptide products are involved, but the interaction of WUS and CLV3 represents a common type of feedback loop that controls many aspects of differentiation and morphogenesis. Figure 14.22. This twig of viburnum was pruned 2 months before the photo was taken. The axillary buds had been completely inhibited by the shoot apical meristem at the time of pruning, but then both became active once pruning stopped the flow of auxin from the shoot apex. The concentration of auxin is greatest at the shoot apex and less at lower levels. At sites where it drops below the threshold level, it can no longer suppress axillary buds, and one or several become active. Other factors must be involved because it is not always the lowest bud that becomes active first, even though it should be the first to encounter sufficiently low auxin concentrations. Botany and beyond. Box 14 to 2 names of genes. Plant scientists working with Arabidopsis thaliana have agreed that the wild-type form of the gene should be written in italicized full caps, whereas any mutant allele is written in italicized lowercase. If the protein itself is discussed, it is in ordinary font. Often, genes are discovered by exposing thousands of seeds to mutagenic chemicals, then sowing the seeds and looking for any aberrant plants with interesting characters. After such a procedure, one plant of A. Thaliana had many more stomata than is typical. That plant was cultivated to maintain the mutated form of the gene. The gene was named after its mutant form, too many mouths, with the newly produced allele being too many mouths. The protein coded by the wild-type gene is too many mouths, and the protein produced by the mutant allele is too many mouths. Table B14, 2 names of genes. Notice that an odd thing about this system is that genes are named after mutant, aberrant characters. The wild-type, ordinary TMM version of the gene produces just the right number of stomata, but its name suggests it produces too many. If the sequence of the TMM allele is known, then the equivalent genes can be searched for in other plants whose development and genomes are well known, for example snapdragon, tobacco, and other plants commonly used in developmental studies. If they are found, then the species initials are used to distinguish them: ATMM, MTMM, NTTMM. These 3 genes all descended from an ancestral TMM gene. The WUS-CLV3 interaction changes both during development of an individual plant and during evolution. In many species, the shoot apical meristem of a germinating seed is very small, but it gradually grows to be a larger size and then stabilizes. By some unknown mechanism, the WUS gene in seedlings is temporarily able to expand its influence and causes a greater number of cells to remain meristematic without triggering CLV3 to inhibit it. When the proper adult size is reached, WUS and CLV3 balance each other and the meristem stops enlarging. Species differ in the typical size of their shoot apical meristems. The typical diameter in Arabidopsis thaliana is about 80 micrometers, but in other species, meristems are as narrow as 50 μm, and 200 micrometers is probably the most common diameter among angiosperms. A few species of cycads and cacti have gigantic shoot apical meristems with diameters up to 2,500 micrometers in diameter. This may mean that during evolution of these species, the WUS gene has evolved such that it is less sensitive to inhibition by the CLV3 gene, or perhaps it is CLV3 that is less sensitive to stimulation by WUS. Of course, it is also possible that other, unknown factors are involved. Figure 14.23 Each cluster of spines on a cactus is an axillary bud, so even though the foliage leaves are too small to be visible, the spine clusters reveal the pattern of leaf initiation by the shoot apical meristem. While this saguaro was very young, its shoot apical meristem was so small and narrow that only a few rows of leaf primordia could fit in the meristem's leaf initiation zone. Consequently, there are only a few rows of spine clusters at the base of the shoot. But as the shoot became older and taller, the meristem increased in diameter so much that there was room for another row of leaf primordia. This happened several times. Arrows indicate where the new rows were initiated. At some point in the future, this shoot's apical meristem will stop enlarging, and after that, the number of ribs will remain constant. A second interaction of hormones occurs in the vascular cambium. Auxin alone activates the cambium and elicits differentiation of xylem, but gibberellin is also present in a healthy stem and causes some of the new cells to differentiate as phloem. Without the interaction of both auxin and gibberellin, a normal, functional vascular system would not develop. Hormones as signals of environmental factors. Leaf abscission. Whereas normal growth of shoots and roots results in large flows of auxin and cytokinin, respectively, environmental factors also influence hormone concentrations. Hormones communicate to various parts of a plant the information that a particular part has encountered an environmental change. Export of ABA by wilted leaves has been mentioned, and another example involves abscission of leaves and fruits. A young leaf produces large amounts of auxin, but production falls to a low but steady level in a mature leaf. As long as auxin flows out through the petiole, activity in the abscission zone is inhibited. If the leaf is damaged by animal feeding or water stress, auxin production drops to such a low level that its flow through the petiole does not keep the abscission zone quiescent. Perception and transduction in this case may be simply that insect or wilt damage makes it impossible for the impaired cells to produce enough auxin to inhibit the abscission. Old age of the leaf may also result in the lack of sufficient auxin, but evidence suggests that autumn conditions stimulate production of ethylene, which then suppresses auxin production and transport in time for abscission before winter. Fruits are prevented from abscising prematurely by the presence and export of sufficient amounts of auxin through the pedicel. Fruit ripening is under the control of both auxin and ethylene, at least in edible, fleshy fruits. Initial transformation of the ovary wall into a fruit is a response to auxin synthesized in developing embryos and transported to the ovary wall. Auxin stimulates many changes, including cell enlargement and differentiation. There is usually surprisingly little cell division during the formation of fruits, even large ones. One effect of auxin is release of ethylene by the developing fruits, which leads to other aspects of ripening in both climacteric and non-climacteric fruits. At maturity, the high concentration of ethylene stimulates the pedicel abscission zone, overriding the presence of auxin. Figure 14.24 A healthy leaf produces and transports enough auxin to suppress activity in the abscission zone. A damaged leaf produces less auxin, insufficient to prevent abscission. Autumn stimuli may cause the production of ethylene, which then suppresses auxin synthesis and transport. An alternative hypothesis postulates that autumn conditions directly suppress auxin production and ethylene is not necessary. Tropisms. Light is the stimulus in phototropism, therefore, a pigment is needed, and its absorption spectrum must match the action spectrum. The action spectrum indicates that blue is the most effective wavelength for phototropism, and the pigment whose absorption spectrum matches that is a small protein, now called phototropin, with two flavin mononucleotides attached as chromophores. Phototropin is a component of a cell's plasma membrane and becomes phosphorylated when exposed to blue light. Blue light stimulates other aspects of plant development also, and together these are called blue light responses. Oat seedlings are often used to study phototropism. They are grown in the dark, and if illuminated briefly with blue light from one side, they bend toward it. Plants grown in darkness do not have chlorophyll, so that pigment is not present to interfere with studies of absorption and action spectra or studies of presentation time and threshold amount of light needed. Oat seedlings have an outermost protective leaf called a coleoptile, which shows a strong positive phototropic response. Oat coleoptiles are the organs studied most often. The site of perception of blue light is the tip of the coleoptile, and if it is covered with an opaque hood, light direction is not detected. Similarly, if the coleoptile tip is cut off before unilateral illumination, the rest of the coleoptile does not perceive the light. We do not know the steps of transduction, but the result is an asymmetric redistribution of auxin. Auxin is transported from one side of the coleoptile to the other. Neither synthesis nor destruction of auxin is affected. After the differential redistribution of auxin is established, the darker side of the coleoptile, or the stem in other plants, receives extra auxin, and thus it grows more rapidly and bends toward the light. When the coleoptile or stem points directly at the light, neither side is brighter nor darker. As a result, differential auxin transport stops, and the organ grows straight ahead. The site of response, where differential growth causes bending, is approximately 5 mm below the coleoptile tip, figure 14 to 25. In positive gravitropism in roots, the root cap acts as the organ of perception. After the lower side of the root is detected, a growth inhibitor is transported to that lower side of the root cap and then into the root, where it slows growth on the lower side. We know almost nothing about how the position of the statoliths is transduced, nor are we certain of the growth inhibitor. Much circumstantial evidence indicates that it is auxin. How can auxin be a growth stimulator in coleoptiles and stems, but an inhibitor in roots? It depends on the concentration and the tissue's sensitivity. Small amounts of auxin stimulate growth, and larger amounts inhibit it. Coleoptiles and stems typically have suboptimal amounts, so redistribution causes the side with more auxin to have a more nearly optimal level, and that side grows faster. Roots are more sensitive to auxin and ordinarily have optimal or excess amounts, therefore, redistribution causes the side with more auxin to have such an excess it is inhibitory. Flowering. Ripeness to flower. Almost all plants must reach a certain age before they can be induced to flower. Only a few species can be induced as seedlings. Annual plants need to be only several weeks old before they become competent to respond to a floral stimulus, but many perennials must be 5 or 10 years old. Beech trees must be approximately 40 years old. Before this time, conditions that should induce flowering have no effect. Virtually nothing is known about the metabolic difference between the juvenile phase, when plants are incapable of being induced to flower, and the adult phase, when they are sensitive to floral stimuli. The conversion from juvenile to adult is called phase change. Figure 14.25. When illuminated from directly above, oat seedlings grow upward. When a young oat seedling is exposed to light from one side, its outermost sheathing leaf, the coleoptile, bends and grows toward the light. If the coleoptile apex is covered or cut away, no response occurs to unilateral illumination, and thus, the tip is the site of perception. If the site of response is covered, bending occurs and the site of response is not involved in perception at all. In dark conditions or with overhead lighting, auxin is transported symmetrically down the coleoptile, causing equal amounts of growth everywhere. With unilateral illumination, auxin is redistributed, with the darker side transporting more auxin than the lighted side, so the darker side grows faster, resulting in curvature. And auxin can be collected in small blocks of agar or other absorptive material and then placed asymmetrically on a decapitated coleoptile. The side receiving auxin grows, but the other side does not. Figure 14.26. And shoots may contain slightly too little auxin for fastest growth. Thus, redistribution in a coleoptile apex caused by unilateral light causes the side with more auxin to have a level that is more nearly optimal, whereas the other side, which receives less, has even poorer growth than before. And roots may contain slightly too much auxin for fastest growth. In root-positive gravitropism, the horizontal position of a root cap causes statoliths to fall to the side of statocytes, causing a downward redistribution of auxin. The lower side receives extra oxygen and becomes even more inhibited, whereas the side that receives none has a more optimal amount and faster growth. Cold temperatures are the stimulus responsible for phase change in biennial plants, and the process is vernalization. The site of perception is the shoot apex itself. If it is cooled while the rest of the plant remains warm, vernalization occurs. However, if the rest of the plant is cooled while a small heater keeps the apex warm, no vernalization occurs. Presentation time is as short as 1 day in some plants. The transduction process is known to require oxygen, carbohydrates as an energy source, and an optimal temperature just above freezing, between 1 degree Celsius and 7 degrees Celsius. Vernalization results in a stable change. If the plants are returned to warm conditions but are not given the floral stimulus of short days and long nights, they continue to grow vegetatively, without flowering, year after year, but they retain their vernalization and flower whenever the floral stimulus is finally given. We know little about what stimulates phase change in perennial plants.